Hello students, we are back again with a new unit this time, and which is a very important unit for your class 12, that is your unit 7. It deals with genetics and evolution. The first chapter which is included under this unit 7 is your principles of inheritance and variation. Now in this chapter, before going into the internal part of this chapter, if we talk about the introduction, the chapter deals with a very important topic of class 12, that is your heredity, genetics and variation. Before we move on to the what are the different parts dealt in this chapter, we must be very much clear about these three terms, that is your genetics, heredity and variation. Now if somebody asks you what is genetics, so genetics is a branch of biology which deals with heredity and variation. Okay, now what is this heredity? Perhaps in your junior class, if you remember, we have read about an organelle present in each cell of our body, and that organelle is called as your nucleus. In this nucleus is present the chromosomes, and the chromosomes contain the genes. Now this genes is the one which is passed on from the parents to that of their offsprings. So the transmission of characters which is passed on from the parents to the offsprings, this transmission of characters, this property or this branch of biology is known as your heredity. So heredity is a branch of biology which deals with the transmission of characters from one generation to the next. And where are these characters present? These characters are present in the chromosomes. And where are these chromosomes present? These chromosomes are present in the nucleus of a cell. Okay, now next term which I used in the beginning along with the genetics and heredity was your variation. Now what is this variation? Variation is the difference of an organism with respect to its parents. That means when the characters are transmitted from the parents to the next generation or to the offsprings, sometimes some minor changes takes place, and it is because of this minor changes that happens during the time of formation of gamete that results into variation in an organism. That means we are similar to our parents, but we are not the identical copies of our parents, isn't it? That means when somebody comes to your house and he or she compares you with your father or your mother, that means you have a eye color same like that of your father, your that is shape of the nose is like that of your mother, your walking style or you know the texture of the hair or the skin color is like that of either of the parents, or sometimes a blending of the two parents. So this comparison, why does it takes place? Because we have some similar characters which we inherit from that of our parents, but we have our own characters as well. Now why this in our own characters which is not the identical characters from that of our parents? That is because of the variation. So genetics is the branch of biology which deals with heredity and variation. Now you know, man has been studying or has been always questioning about how does character appears in an organism, or how these characters are transmitted from one generation to the next from the time immemorial. If you talk about man being involved in the process of this genetics, this interest grew in man when man first started domesticating the animals, or when man first started doing agriculture. So when man started doing agriculture, that time he actually did various kinds of hybridization experiments, unknowingly of course, to improve the crop variety, because that time the motto was to improve the crop production, to improve the food grains, quality of the food grains, the quantity of the food grains and all these things. And it is perhaps because of this reason of the hybridization that the domestication of the animals also was taken into consideration, and man had successfully being able to find a new breed of a cow, an indigenous cow, which is in India, of course, that is your Sahiwal cow. Now this Sahiwal cow was developed as a result of the various kinds of this breeding experiments which man had been trying on from many years. So it is not that man was not knowing about genetics, but what is the principle behind it, or what things are actually responsible for the transmission of the characters, that man was not knowing until it was brought into that is your to everybody's consciousness by a Austrian monk or a scientist. The name of the scientist was Gregor John Mendel. Now Gregor John Mendel was an Austrian monk. He belonged to a very poor family, but as he was very intelligent from his childhood days in mathematics and science, so he got an opportunity to study these subjects well, and he was allowed to teach to the students, the high school students in Austria after completion of his education. And he was living in a monastery, and there he had a small garden in which he did various kinds of hybridization experiments. In this hybridization experiments, he tried to do the experiments with various kinds of plants, but he got attracted towards a plant that was a pea plant, Pisum sativum. The reason was because this plant was annual, and it was easy to raise crops very soon, and it has some noticeable characters, and these characters were the one which had attracted him. The flowers were quite big to handle with, because when he did the hybridization experiment, he could handle the flower well, because the flower was large in size, and also the flower was bisexual, so it could also be used for the self-pollination process, and it could also be used for the cross-pollination purpose. So self-pollination, why? Because when Mendel wanted to learn about this pea plant, or about the things which are getting transmitted from one generation to another, he wanted a pure plant. So for this he actually did selfing of the plant for various generations, and after he was assured that the character for which he is working for many generations, it was pure line, then he could assign, or he could find out that the pea plant had 7 pairs of contrasting characters, and these characters he noted down. Since he was a mathematician, so he actually tried to explain the biological phenomenon with respect to mathematics. Now this was perhaps also one of the reason why his experiments, which he had done, which he has calculated, which he has meticulously done, written all the observations years after years. He did 7 years of long experiments on this pea plant, and came into the conclusions, that is the principles of the inheritance, and he wrote it in a journal, and this was published in a journal which was very obscure, which was not so renowned journal. So that was perhaps the reason why his work was unnoticed, and it was only after his death, after 16 years of his death. He died in 1884, and after 16 years of his death, that is in 1900, when 3 different scientists from 3 different corners of the world came to the same conclusion which Mendel had done during the time of his experiment with the experimental material, that is the pea plant. So this was the utter disappointment, because Mendel worked so hard, people did not notice. During that time, people were more acknowledging Darwin for his theory of evolution, and Mendel's work was ahead of his time. People did not or could not relate mathematics with that of biology. So this was perhaps the reason why his work was unnoticed. But after his death, when his work was again rediscovered, and people found that what a great work this man has done or contributed towards the field of genetics, he was given the title of father of genetics. So it was pathetic. During his lifetime, he could not get the kind of recognition of his work which he had done, but after his death he was given this honor of father of genetics. So that was the story of Mendel, and why did Mendel selected the plant material as pea plant. Some people are of the opinion that Mendel selected the pea plant or rejected the experimental results which were hindering with his principles. But that is again a question mark. We are not going for that. But he was successful with all the 7 pairs of contrasting characters which he had studied in the pea plant. Now these contrasting characters are what he did not know that time, any genes and all, because that time no microscopes were there. So he named this which are transmitted from one generation to the next generation as factors. Now these factors are now presently known as your genes. Now these genes which he talked about, each gene has 2 versions, or two forms rather. Now these 2 forms are called the alleles, allelomorphs, or it is called as your alleles. Now what are these? Suppose the gene responsible for the character of height. Okay, so this is located in the chromosome in the same locus. That means if we have because chromosomes are always in pair, homologous chromosomes, isn't it? So when they are in pairs, so the location of suppose a gene and the next, that is the homologous pair, so let us make it as A dash. So A and A dash gene will be in the same place, and these 2 versions of the gene are can be same, can be different, can be in dominant form, can be in recessive form. So when they are same, when the 2 forms of the same gene are in the same, that is they are of same type, then we call it to be the homozygous condition of that, that is your particular character. Like for example, if we talk about Mendel's say the height of the plant, that is tallness, if we represent it with the alphabet capital T, then capital T represents the dominant nature, homozygous nature of the height of the plant. That means both the alleles present in the chromosomes represents the same type of allele. They are similar, they are same, okay, identical. But if suppose the 2 alleles of the same gene are different, one is capital T and another one is small t, in that case it will be called as your heterozygous condition. Heterozygous condition, why? Because here you see the 2 alleles, the 2 forms of the same gene are not same. One is dominant, that is capital T, and the recessive one is small t. Okay, expression may be same like that of capital T, but if you talk in terms of the gene expression, that is a genotype is not same, isn't it? So we call it to be the heterozygous condition. The former was homozygous, that is capital T, when both the alleles are identical in the of the same gene, and when both the alleles are different of the same gene, then we call it as the heterozygous condition or the hybrid condition, as for example capital T and small t. I am talking about genotype, not phenotype. Phenotype means the outward expression of the organism, the physical expression of the organism. Physical expression, both the cases it will be tall, but genetic expression, if you talk about genotype, if you talk about, the former is homozygous and the latter one is called the heterozygous condition. Now there is a third form also. The third form is what? The recessive condition, homozygous condition. That means both the forms of the gene are same, but they are of recessive form, that is small t. So there are 3 forms. That means one is dominant form, which is that is homozygous dominant, another one is heterozygous, and another one is homozygous recessive form. So this was the 3 possible forms or versions of the gene. Now the characters which he selected, 7 pairs of contrasting characters. One was with respect to height, that is tall and dwarf. Then was flower color, that is purple and white. Then it was position of the flower, axial and terminal. Then it was the shape of the seed, that means round or wrinkle. The color of the seed, yellow and green. Then shape of the pod, inflated or constricted. Then that is your color of the pod, yellow and green. So these were 7 pairs of contrasting characters were taken into consideration, and each time he crossed them. What he did at first is that he did self-pollination. See, when the pollination experiment is done, you have read it in the sexual reproduction of flowering plants that there is a process when artificial hybridization is done. In case of plants, we if it is a bisexual flower, we cut the anthers before it becomes mature. That is called emasculation. So he emasculated the flower because he did if he want to do the cross pollination, but when he did the self pollination, he did not do so that it does not get contaminated. Suppose he got a pea plant with a character in which he was studying is tallness, so he tried to do the self pollination by covering the inflorescence of the flower with the help of a polythene bag. And when the self pollination took place, after that, when the seed resulted, formation of seed happened, then he took or he collected these seeds of the self pollination, and then he again sowed them in his monastery garden, and then he found that again the next generation were tall. So this way he repeatedly did this experiment and again, and he now, when he was sure that this plant is only going to show tall character, then he labeled it as the pure line for the tall character. Similarly, he did for the other 6 pairs of contrasting characters, and he made this separate observations noted down, and he named it as purebred for that particular character. And then after he was sure, then he took the 2 pure forms of the height character, that is tallness and dwarfness, and then he cross pollinated. So when he cross pollinated the first generation, which is called the first filial generation, he observed that all the organisms of the first generations had only one dominant character, and that was the tall character. After that, he wanted to know that what happened to the next character, because parents were 2, isn't it? One was tall and another one was dwarf. What happened to the second parental character? Where has it disappeared? So what he did is that when he obtained the seeds of the first generation, he again grew them in his garden. The F1 generation seeds, he grew them in his garden, and then he again pollinated them, of course the cross pollination, and he found the F2 generation plants, the one which were growing in the F2 generation, they were showing a reappearance of the second parental character which disappeared in the first generation. That means along with the tall plants, he observed that some small section of the population were showing the characteristics of the dwarf parent as well, and he counted them. And since he was a mathematician, so he took a ratio, and he found that the ratio was coming 75% and 25%. That means 3:1. That means 75% of the population were showing the tallness character and 25% were showing the dwarf character. So he wanted to assure whether this experimental observation which he has got is also similar with that of the other 6 pairs of contrasting characters. So he took all those 6 pairs of contrasting characters as well as the pure line breed. He crossed them regarding the flower color, regarding the position of the flower, regarding the seed color, everything, and to his observation, he found that out of the pair of contrasting characters, always one was dominant over the other, and the second parental character did not appear in the first generation, but it could reappear again in the F2 generation. So from this he postulated his principle of inheritance, or which he postulated his first law of inheritance, you can say, which he named it as law of dominance. And according to this law, out of a pair of contrasting characters, one appears in the first generation, other remains hidden, and this hidden character which is not found in the F1 generation again reappears in the F2 generation. Mendel was lucky that there was no blending of character. No blending of character, no intermediate character he observed. Later on, when the scientist performed various experiments, depending upon his observation, blending character was found. We will discuss it later on, but for the time being, we are concentrating upon the Mendel. So this was the law of dominance, and also Mendel said one more thing which is called the law of segregation, that when the gametes are formed as a result of meiosis, isn't it? That means half the number of the chromosomes. He did not know that time chromosomes, what are genes and all those things, but according to the observations he had undergone with the pea plant, he also postulated that is the law of segregation, which is also known as the law of purity of gametes. That is, during the gamete formation, the characters separate, the alleles they separate, and they again when they combine, or when they fuse, the two separate characters, when they fuse in the zygote, that means a new organism is formed. And now in this, the one which is dominant will express itself, and the one which is recessive can express only in homozygous condition. It cannot express itself in the presence of a dominant character. So that was the law of segregation which was proposed by Mendel. So today we have discussed about the Mendel's life, his work, his contribution towards the field of genetics. What is genetics? What is heredity? What is variation? What was his law of dominance? What is the law, that is your law of segregation? And why did Mendel selected pea plant for his experiment? And what was done later on, and why his work was unnoticed, and all those things. I hope you understood the introduction part of this chapter well. We will take upon the next part of this chapter again in the next class to come.